Hello, everyone, and welcome to the third live session within the third week of the first International Electronic Conference on Genes, Theoretical and Applied Genomics. This conference has been organized by MDPI Genes Journal, and is being chaired by Dr. Aureliano Bombarelli from the University of Milan in Italy. I would like to say thank you to our chair and the conference committee for making this conference possible, and all the participants for joining us today. Just as a reminder, until Monday, 30th of November, you can read and comment all the author submissions presented for the conference, and you can also enjoy some recorded and live sessions. Please look at our webpage during this month for more updates and register for free for the following live sessions. I would like to remind also everyone that this webinar is being recorded and that we will have YouTube live streaming today. The recording will be posted on the conference webpage for everyone to watch it as soon as possible. The speaker for this session is Professor Michael Hofreiter from the University of Potsdam in Germany. He completed his PhD in biology in the Max Planck Institute for Evolutionary Anthropology, Leipzig University in Germany. After some postdocs and being a professor for ecology and evolutionary biology at the University of York, he is now a professor for ecology and evolutionary biology in Potsdam. His research interest lies in elucidating the genetic basis of animal adaptations and the effect of environmental changes on both neutral and adaptive genetic diversity. At the end of his talk, we will have a Q&A session, so if you have any questions, please feel free to post them in the Q&A box at any time. So that's all from my part. I will hand out now the microphone to him. Thank you, Michael, for accepting to participate in this session, and everyone enjoy the talk.
Okay, thank you very much for the introduction. So let's see how this works. Okay, this is the first lecture in that case or presentation I give online. So it's an interesting experience. So what I'll talk today about today is a paleogenomic perspective on evolution, looking at lost lineages and sometimes also ghost lineages. So what it is about getting DNA sequences in relatively big amounts, not like in the old days where we had short sequences, but full genomes from bones that are sometimes thousands, sometimes tens of thousands, and sometimes even hundreds of thousands years old. What I'm not going to talk about, unfortunately, is dinosaurs, because dinosaurs are simply too old. So we haven't managed yet, and almost certainly will never manage to get any DNA from dinosaurs, which is a shame, but nothing we can change. The time frame for ancient DNA, I'll give a little bit of an introduction on ancient DNA before I go into the biological questions, is roughly shown here. And it depends very much on the temperature the fossils come from. So in permafrost, you have much longer survival of DNA. At the moment, around 700,000 years. But I think it is fair to say that the time frame will extend a little bit in the permafrost and possibly, or almost certainly, reach around 1 million years, which is still far away from the 65 million years we would need for dinosaurs. Outside the permafrost, it is of course shorter, and the published age at the moment is 400,000 years, and there's probably not going to change so much in the near future. So roughly 400,000 years outside the permafrost. Maybe we're going to reach 500,000 years there, and in the permafrost, 700,000 at the moment, and we're likely going to reach 1 million years, which is actually not that bad if one looks at the fact that the Pleistocene ended only 11,000 years ago, and human civilization is even younger. And what you can also see is that this covers a couple of climatic fluctuations during the Pleistocene with warm times and relatively cold times. In Potsdam, in my group, we work on many different species, not only extinct ones, also extant ones. But if you look at them from the perspective of a geneticist or genomicist, they pretty all much reduce to 4 letters: A, T, G, and C. So it's fair to say that for a geneticist, life is in some ways relatively simple. Unfortunately, if working in ancient DNA, there are a couple of things that also complicate life. And one of these problems is the properties of ancient DNA or rather DNA in general, because if you take away the proteins that allow 1.5 meters of DNA being folded in our body cells at this tiny space, if you take these proteins away, DNA becomes a rather stiff and fragile molecule. I always compare it a little bit to a 10-kilometer long raw spaghetti. And if you want to have a picture for ancient DNA is basically what you would get if you would throw the 10-kilometer long raw spaghetti from the Eiffel Tower. In other words, it's short. Ancient DNA is fragmented to a tremendous amount. So we have samples, admittedly, these are the worst samples we have, where the average read length is about 30 base pairs long. So for a normal mammalian genome to get assembled from such pieces, you need 100 million pieces. So it's a rather big jigsaw puzzle. Problem is also, of course, such short fragments, even with a computer, are difficult to put together because a 30 base pair fragment doesn't contain so much information. We don't use fragments that are shorter than 30 base pairs, but as you can see here, very few, almost no fragments actually exceed 100 base pairs in length. So we're really dealing with the short stuff. And we mostly work from cold or temperate environments, which is shown in a bit complicated way in this figure. What you see in the top panels is a picture for DNA preservation, not on the surface, but below the surface, where temperatures are colder and more constant. And at the bottom, you see DNA preservation on the surface. And on the left side, the left panels are for 150 base pair pieces, so survival of 150 base pair pieces. And on the right side, 25 base pair pieces. And in red, poor survival, in blue, good survival. And what you can actually see is there's not that much of a difference, but it's slightly better, or it's better for shorter pieces, and it's slightly better below the surface than on the surface. And if you look, for example, on the surface in the left lower panel, for 150 base pairs, practically all of Africa is red. So no DNA survival in Africa or Australia and most parts of Asia. If you go to the upper right panel with 25 base pairs below the surface, you can see that the most southern parts of Africa and Australia, we have some DNA survival. And you can also see that in Europe, we also have some DNA survival in those cases. So the colder, basically the better, which is in a way a shame because many interesting species live in warmer areas, but the tropics are mostly out of reach for ancient DNA, especially if we go to the older times. So how do we get ancient DNA? Not like fossils, like is shown here, but basically we get them from the fossils. We grind parts of the fossils. We don't need very much, actually. Modern ancient DNA work can start from 20 milligrams or 50 milligrams of bone. Sometimes we use even less. So we don't need big chunks of fossils. And we grind those small pieces. We dissolve them in a buffer, and then we purify the DNA. And I won't go into more details about the technology, except for one point, because that is important. and that is the choice of bone, because there is one bone in the mammalian skeleton. My group mostly works on mammals, that absolutely exceeds in DNA preservation, and many studies have now confirmed this observation that was first published in 2014, and that is the so-called petrous bone, part of the inner ear. And what you can see with the errors is roughly not 100% correct where you find the petrous bone. And on the left, with the circles, you can see what you have in terms of endogenous DNA in black in a typical bone, like if you take a long bone and cut out a piece. And you can see that the endogenous DNA content, so the DNA from the organism the bone derives from, is actually a tiny part of the total DNA. And when it's a sample contamination, it actually doesn't mean human contamination. That also happens, but it's not very common. Well, it is, that's wrong. It's actually common, but it usually doesn't make a large share of the DNA. What you really get mostly with most bone types is microbial DNA. So bacteria, fungi, and maybe archaebacteria that have been living on and from the bone, and only a tiny part is really what you're interested in. For the petrous bone, it's different because often 50% or even 75% of the DNA from a petrous bone belongs to the animal the bone comes from. Now, this is, of course, great for genome sequencing because it means you have to spend much less money to get a certain amount of data, even though it doesn't solve the problem that the DNA is short. So the DNA is also fragmented in the petrous bone, but the percentage of DNA that is usable is much higher than for most other bones. There's some ongoing work to make other bones usable or better usable. That's very promising, but I'm not going to go into details. We can, of course, treat that in the discussion. So now let's get to the point, what is one doing with ancient DNA as an evolutionary biologist? And of course, the picture has nothing to do with ancient DNA, but since ancient DNA researchers work with rather dull and gray bones, I wanted to have at least one nice biological picture of beautiful looking animals. And coral reefs are also an excellent example for evolution or for the process of evolution. So I think it fits well to start with the biology. What I will talk about is bear evolution, and specifically for a certain group of bears that you can see here, 3 species: the polar bear, the brown bear, and at the bottom, because it's extinct, we only have a drawing rather than a photograph, the cave bear. We are mostly interested in the cave bear, but the cave bear is the closest relative of brown bears and polar bears, so naturally we use them, these 2 extant species, for comparison and for investigating the evolution of cave bears. And as it turned out, we also learn a lot about the evolution of the other species that way. The work on cave bears was mostly done by Axel Barlow, who is shown here, who was a postdoc in my group and is now at the University of Nottingham, and normally works with venomous animals like this. I think it's a Chiller monster. He also works a lot on venomous snakes, but I could convince him to work for a couple of years on harmless and dead animals, mostly on the cave bear. So cave bears obviously are extinct. So normally we would treat them as a lost lineage. But the question, it turned out, when we look deeper into the genomes of the species is, are cave bears really lost lineage, or are they rather ghost lineage? So has something survived of the cave bear? To investigate that question, we didn't travel to the Caucasus, although it's a beautiful place. but we got samples from our collaborators, in that case, Gennady Baryshnikov from Russia, from the Caucasus. And one of the samples is shown here for a special reason, because it comes from a layer that was dated to approximately 360,000 years. So for ancient DNA outside the permafrost, this is rather old. Even so, very little data has been published from samples that are that old, but basically almost no genomic data. Mitochondrial DNA, but very little nuclear DNA. And that is the bone, that is the petrous, and some associated bone around it. Now, cave bears are something with complicated evolutionary history. So there's a number of taxa that are variously listed as species or subspecies or variants, and a few of them, not all of them, are shown here. The Caucasus cave bears are put into two different taxa. The younger ones are called Kudurensis, shown in orange, and they have this typically domed forehead that is typical for all late Pleistocene or most late Pleistocene cave bears. And the older ones are called Pre-Kudurensis. They are earlier. They are shown in yellow, and you can see that the forehead is less domed. They're more similar, actually, to brown bears. And there are a couple of other taxa you find in Europe, and you also find in the Urals. By now, we know that we also find cave bears even from Siberia. So they have a pretty wide distribution, but usually only you find them when you have karst areas. So you have many caves, which gives the name. I should mention that actually cave bears, of course, didn't live in caves, but we find the skeletons in caves because they were hibernating there and often died during hibernation. So we have large assemblies of bones. It might be, together with the mammoths and the steppe bison, the Pleistocene extinct species with the largest fossil record. So when we looked at the mitochondrial relationships, you can see a picture here. We found the Caucasus bears as an outgroup to all other cave bears. The Pre-Kudurensis is shown in smaller size because they were actually smaller. The Kudurensis were relatively large. You can see the arrow to the Denincheri, which is a Spanish cave bear, also relatively old. And then you have a couple of other groups from partially the Urals, like Rossicus, and Europe, the other three, the blue, green, and red taxa. And this is a phylogeny or a topology of the tree we have been working with for a long time, because for a long time we didn't have anything else than mitochondrial DNA. Now, the interesting thing when we got nuclear DNA was, or one of the interesting things, that the nuclear tree doesn't agree at all with the mitochondrial tree. In fact, there are only a few nodes that agree between the two trees, and basically it's a split into the two big groups, the European Uralian subgroup and the Caucasian, and also the topology within the Caucasian bears. But within the European Uralian group, actually not a single node agrees between the nuclear and the mitochondrial tree. That is an extreme version of the brown bear polar bear tree, because in brown bears and polar bears, it's also the case that the nuclear tree treats brown and polar bears as reciprocally monophyletic groups, whereas in the mitochondrial tree, the brown bears are paraphyletic compared to the polar bears, meaning that some polar bears are more closely related with brown bears than these brown bears are with other brown bears, as you can see on the right side of the tree. In brown bears and polar bears, this is explained by mitochondrial DNA transfer, mitochondrial capture, and it looks from the tree that this process was even more abundant within cave bears. So we get a lot of mitochondrial transfer between the different cave bear populations. Another interesting aspect of this is that, as in brown bears and polar bears, in cave bears also the nuclear tree agrees much better with the morphological tree than the mitochondrial tree. So we had lots of discussions with the paleontologists who weren't happy with our mitochondrial trees until we got the nuclear DNA. And the nuclear DNA now fits perfectly with the morphological tree, which in hindsight maybe is not surprising because morphology is mostly determined by the nuclear genome. But nevertheless, it made our life in some ways easier because it shows that the mitochondrial DNA is the outlier. It also shows, which is not shown here in the figure, that large body size in cave bears probably only evolved once because all the large-sized lineages, which are the Speleos, Ingressus, and Carnivets, fall together in the nuclear tree, whereas they are spread out in the mitochondrial tree. Now, we can also look at the nuclear relationships in a simple PCA, and then we basically see the same picture like with the tree, just a bit clearer. We have three large groups. One are the Caucasian ones, one are the European ones, and then we have the so-called Rossicus from the Urals that are clearly spread out in the tree, in the PCA, and you can also see that in the tree here, these are the three main lineages. What we can also do with the age of this Caucasian Precoturensis with almost 400,000 years is actually estimate the substitution rate. And we did that with a lot of cross-testing, and it works surprisingly well because it's so old. It gives us a direct estimate how fast the nuclear genome, and also it works also for the mitochondrial genome, evolve on a molecular level. And with this substitution rate, directly estimated substitution rate, we can actually date our tree, and that gives a quite interesting result. It's actually shown here. And what you find first is that cave bears and brown bears and polar bears separated exactly when it was predicted by paleontologists about 1.6 million years ago. And what you can see with all the black spots are also different estimates with the different pairings we have between brown bears and cave bears. And you can see that they all fall in a quite narrow range. And of course, we can date the other nodes. And you find a number of nodes, the separation of the main lineages in the cave bears, the separation of polar bears and brown bears, and even the separation of the deepest brown bear lineages, all within a relatively narrow time frame, around 1 million years, maybe 800,000 to 1 million years. So we have several major divergence events around this time. And the question, of course, is this coincidence or did something happen there? And also with a single species group, we can't make a strong case. It is interesting to see that around 1 million years ago, the Pleistocene climate cycle changed from a 41,000 year cycle between the climatic fluctuations, so too cold or too warm times, to 100,000 year cycle. And with this change in cycle lengths, also the cycle amplitude increased, and it seemed that this change to longer and more dramatic fluctuations in temperature might actually have caused the divergence of the different bear lineages. Of course, it would be nice to look in other species if we find the same pattern. What we also did with the data is to look at gene flow. It is known that there was gene flow between brown bears and polar bears, and we were interested in looking whether there was any gene flow between cave bears and brown bears, possibly also polar bears. Also, so there's no evidence that they all ever lived in the same habitat, but brown bears and cave bears definitely occurred in the same habitat. And the standard test for this is t-statistics, and we actually found no matter which comparison we used, so we used a couple of cave bears compared to the list of brown bears that's shown here, the t-statistics was always significant. Now, a bit of a problem with these statistics is it doesn't tell you which directions the gene flow occurs. And therefore, Axel devised a new test, a phylogenetic test, where he divided the genome in windows and then used a 4-taxon tree, 2 cave bears, brown bear, and polar bear, and collected the events where he got trees that disagree with the overall genomic tree, which is cave bears as sister, the 2 cave bears as sister group, and brown bear and polar bear as sister group. And if we only had something like incomplete lineage sorting, we should find the brown bear as together with the cave bears to the exclusion of the polar bear as often as the polar bear together with the cave bears to the exclusion of the brown bear. However, that is not the case. We find many more trees where the brown bear falls together with the 2 cave bears to the exclusion of the polar bears. And we can actually use that as evidence that gene flow has taken place from cave bears into brown bears. We can also use the other type where a more admixed cave bear falls together with brown and polar bear as evidence for gene flow in the other direction. And however, there you can see that the difference between the number of events is much smaller. So if you look at class 3 and class 4, there is little difference, a little bit, yes, but if you compare class 1 and class 2, the difference is massive. And using the number of occurrences, we can estimate actually a percentage of admixture. And we found that modern brown bears are admixed between 2 and 4, well, actually 2 and 3%. The 4% admixed brown bears, the Pleistocene brown bear from Austria, with cave bear. So we have cave bear variability surviving in brown bears, not in polar bears. And we also had a little bit of brown bear DNA in cave bears, but not as much as the other way around. So the admixture was rather unidirectional, not completely, but mostly from cave bears into brown bears. And actually some of the genetic diversity of cave bears survives in modern brown bears, like some Neanderthal diversity survives in modern humans. We can also date the timing of the admixture if we compare which lineages are admixed the same way or to the same amount. And we find that the admixture from brown bears into cave bears actually took place in a rather narrow window between the divergence of the major cave bear lineages, the Caucasian cave bears from all others, and the divergence of all other lineages. So around 1 million years ago. So at the same time when we had the divergence of the major evolutionary lineage, we also had this admixture taking place. We also have another admixture event in cave bears between the Caucasian ones and the European ones, but we can't date that as well because we have this long unbroken branch. So in a way, cave bears as a species are lost, but they actually do survive in brown bears as a ghost lineage, even they don't look as horrible as this one. Okay, we can also look at gene flow within cave bears, and I saw I have to speed up a little bit because this is going to take too long otherwise. And there's an interesting case in cave bears we find in Germany, because we have these 2 separate mitochondrial lineages. The mitochondrial tree is shown here on the right, that usually don't occur together. So the blue lineage mostly occurs in Western Europe, the red lineage mostly in Eastern Europe. But we have a place in Germany that's called the Aach Valley, where they actually occur in the same caves. And as you can see in the lower panel, they overlap, but they don't totally intermingle in time. So the blue lineage mostly occurs in the older layers, and the red lineage comes in, and before both go extinct, it eventually replaces the blue lineage. But of course, this is only mitochondrial DNA. So we looked at genomic nuclear DNA and found that on a genomic level, you can't distinguish them. So you have cave bears from different places, from Romania, Austria, Spain, and Germany. And you can see that the German ones, independently of their mitochondrial DNA, fall together with the Spanish one that also has, of course, a Western European mitochondrial type. It's slightly different in Romania, and the two places will come together in a minute, because in Romania, when we look at different caves, we find the cave bears, which are shown, the oldest one in blue, the middle-aged and in orange, and the youngest one in purple. You find them in different places in the PCA, but it actually doesn't follow a locality pattern. It follows a temporal pattern. So we have three populations in Romania of cave bears that are spaced in time rather than in space. And when we looked a bit closer into that, we found that when we look at the old Romanian ones, they actually are rather distinct from the middle Romanian ones. But the middle Romanian ones are kind of continuous with the younger ones. At least we have definitely some genetic continuity, but at the same time, we do have gene flow, nuclear gene flow from Germany and Spain. So even so, in Germany, we find the Eastern lineage, the Eastern mitochondrial lineage occurring at some point. We find in Romania on the nuclear level, Western allelic variability showing up. So the nuclear gene flow goes in the opposite direction to the mitochondrial gene flow. Which is a quite remarkable pattern, which I think hasn't been observed yet with ancient DNA. So it's summarized here in this relatively complicated picture. With the old Romanian ones, this population probably went extinct, and it looks like they went extinct always the populations around Heinrich events. So these are colder phases of the Pleistocene, and they were stable in the warmer phases, as you can see here. And the middle Romanian ones seem to have come from a population that's closely related to an Austrian population. They are continuous with the younger population, but with input from a population, from a Western population that we find in Germany and Spain. And actually in Germany, we find a continuation on the nuclear level, but a replacement on the mitochondrial level from the east. So we have nuclear gene flow from west to east, and mitochondrial gene flow, interestingly, in the opposite direction from east to west. Okay, so to conclude on the cave bears, part of the cave bear genome has survived in modern brown bears, and it shows that physical extinction not necessarily means that evolution has stopped for this lineage. Single locus data, we have long known that, but they give really an incomplete picture of evolutionary events, so they should be treated with caution. Gene flow as well as population structure happen constantly, or changes in population structure happen constantly throughout space and time. And gene flow sometimes may run counter to the dispersal direction, or at least it looks like it runs counter to the dispersal direction when we look at mitochondrial DNA compared to nuclear. And if our samples are old enough, we can directly estimate substitution rates and that way date divergence events. So much for cave bears. Now I'll try to quickly come to two other species groups, and we'll start with cave hyenas. And what you can see, of course, is not a cave hyena, but a close relative, the modern spotted hyena in a zoo in Scandinavia. So they actually feel reasonably happy when it's cold. And that work was done by Mick Westbury, who was a PhD student in my lab and is now in Copenhagen. Now, spotted hyenas of the genus Crocuta only exist in Africa these days, but they occurred across most of Eurasia and also north of the Sahara during the Pleistocene, though they had a much wider distribution. In 2005, we already did a study only based on mitochondrial DNA, found two clades in Africa, and one of these clades also in Eurasia, but two more clades in Eurasia, one restricted as far as you know to Europe, and the other one restricted to East Asia. And based on the distribution of these sequences and the timing we inferred from the tree, we came up with a scenario that we had three dispersal events from Africa, one less than 3.5 million years ago, the other one around 1 million years ago, and a relatively recent one within the last couple of 100,000 years. Several years later, another group came up with an alternative interpretation, and they argued that the divergence is much younger and that actually the spotted hyena and cave hyenas had an origin in Eurasia, and that they dispersed from Eurasia to the different places. And what we see is mostly relatively recent lineage sorting. Now, both studies were based on very little data, short fragments of mitochondrial DNA. So the simplest way to resolve that is getting more data. And that's what we did in 2018. We resampled and we got genomic data. Of course, we had fewer samples, but we had reasonable sampling in Africa and relatively patchy sampling in Eurasia, because very few of the samples were good enough for genome sequencing. But we had some East Asian ones and some European ones. So what did we find? With the mitochondrial DNA, we found a very similar tree to the previous one. We found the deep divergence in East Asia. We found purely African haplogroup, and we found the purely European haplogroup, haplogroup C, and then the shared one between Europe and Africa. And also the dating was roughly the same, relatively ancient. And if you look at that tree, it looks actually quite similar to the human tree if you include Neanderthals and Denisovans, a very deep divergence of the East Asian group and a less deep divergence of the European group, haplogroup B. However, the nuclear tree looked different as in humans, because for the nuclear tree, we found cave hyenas to be monophyletic and spotted hyenas in Africa to be monophyletic. And within cave hyenas, we found the divergence between Asia and Europe. So slightly different tree. And again, nuclear tree doesn't fit with the mitochondrial tree. Now, what we then also did is we looked if we found evidence of gene flow, and as in the cave bears, we did find evidence of gene flow, and we did another analysis. We looked at the fragment lengths of the fragments that were transferred. And what you can see here is how many consecutive 100 kb blocks we found that disagree with the species tree, so that are basically either introgressed or the result of incomplete lineage sorting. Now, the least evidence for either of the two processes we find in the East Asian cave hyenas, that's the red line. And as you can see, that's also the oldest because it's the shortest fragment. 95% of the fragments are just in one block. For the next event, and that must be gene flow, we find introgression in both directions because we find cave hyena segments in the Namibian hyena, and we find spotted hyena segments in a German hyena, that's a blue line. And here the fragment size is basically identical. So we can expect that this was one event where we had bidirectional gene flow really. So we find from this gene flow event, we still find some traces in of cave hyena DNA in the spotted hyenas. And we also got some DNA transferred from spotted hyenas into cave hyenas. And then we had another much younger event because you can see there's only 85% are in a single block, and a considerable amount, about 10%, are in blocks of 200 kb, and then there's still almost 5% left in even longer blocks. And that was another gene flow event from cave hyenas. into spotted hyenas, and we find that especially in the Kenyan, so in the East African hyenas, which also makes geographically sense that this one's younger. So this tree is not to scale, but what we see are the two gene flow events, one bidirectional, a younger one from Europe into Africa. So we have substantial gene flow, substantial survival of cave hyena DNA in spotted hyenas. And what we can also see is that the divergence between the two lineages based on the nuclear dating is incredibly old, around 2.5 million years. So these are deeply diverging lineages, much deeper than humans and Neanderthals, and even deeper than cave bears and brown bears. Nevertheless, morphologically, they are, as far as we can tell from the bones, very, very similar. So spotted and cave hyenas really have a relatively ancient divergence compared to what we look at otherwise, even more ancient than brown bears and cave bears. There's a discordance between mitochondrial and nuclear tree that is, funnily enough, similar to that we find in humans, Neanderthals, and Denisovans, although I would argue that's simply a coincidence. We find evidence for multiple gene flow events between Africa and Eurasia, and actually the last one must have been around 500,000 years, so around 2 million years after the initial divergence. So even mammalian lineages are able to exchange DNA quite long after the initial divergence. And like with the cave bears, cave hyenas survive as a ghost lineage in modern hyenas. That brings me to my last example, and that comes from elephants. And what is shown here is Paleoloxodon antiquus, the straight-tusked elephant that lived in Europe until 120,000 years ago. And in some patches, it might even have survived until 30,000 years ago, but in dwindling numbers and eventually went extinct. It was one of the largest mammals ever, with a shoulder height of about 4 meters and a weight estimated up to 13 tons. So substantially bigger than our modern elephants. That work was done by Sina Baleka, another PhD student in my lab. And you can see here standing behind a femur of such a straight-tusked elephant. And even so, the femur looks bigger, of course, because it's in the foreground. You can see that these animals had pretty massive bones. Unfortunately, this femur didn't yield any DNA. It's from the North Sea. However, from a site in Germany, Neumarkt Nord, and another one, Weimar Ehringsdorf, shown here in orange and blue, we did get initially mitochondrial DNA, and that already gave the first funny result, because the mitochondrial DNA for these elephants is dispersed among the mitochondrial genomes of the African forest elephant, which is the place where they shouldn't be. In fact, based on morphology, paleontologists argued that Paleoloxodon belongs to the genus Elephas, the Asian elephant, and is quite closely related to the modern Asian elephant. Now, the place of the mitochondrial DNA doesn't fit at all to this, but the two groups of fossils were done independently in two different labs, so it seems rather unlikely it's an artifact. Moreover, we then did some additional sequencing of a Sicilian dwarf elephant, which is shown on the right. It wasn't as small as the smallest dwarf elephants from the Mediterranean islands, which is shown on the left, which had a shoulder height of about 1 meter, but it was quite small, with a shoulder height of about 1 meter and 70 or 80, and a weight of approximately 1 ton. And it's supposed to be derived from the giant straight-tusked elephant that's shown in outline at the back. Now, Sicily is, of course, a beautiful place, but as you remember, warm temperatures are not ideal for DNA preservation. So we had to screen quite a number of petrous bones until we eventually found one that was just good enough to retrieve a mitochondrial genome. Unfortunately, basically no nuclear DNA. However, with the mitochondrial DNA, we could do a tree, and what one can't see too well here is in the upper panel, the position of our Sicilian elephant is shown in red. So you have basically at the bottom, you have the mammoths, then you have the Asian elephants, then you have the three sequences of Neumarkt Nord, the straight-tusked elephant, and then in red you can see our Sicilian elephant. So actually our Sicilian dwarf elephant is on the mitochondrial level more closely related to the Neumarkt Nord, the 120,000-year-old elephant, straight-tusked elephants from the mainland, then the other straight-tusked elephants from the mainland, from Weimar-Ehringsdorf, which are actually much older, about 200,000 years, are two of these big ones. So the two big populations of straight-tusked elephant are more distant in the tree than the dwarf elephant is to one of these two populations. Now, what we then, of course, were interested in is how fast can such a huge animal actually shrink? And for that, we did a data tree and estimated the divergence time to get an estimate of the dwarfing rate. On the left, you can actually see Sicily with different places, and the sample was from the north of Sicily in Puntali, which is shown with a small square. Now, what we found is that the maximum divergence or the divergence between the Neumarkt Nord elephants and the Sicilian dwarf elephants is around 550,000 years ago. Probably they colonized the island later, because I mean, the genetic divergence for sure predates the population divergence. So with the genetic divergence, we get a minimum dwarfing rate, and with the fossil record, we can get actually a maximum dwarfing rate. And with these two brackets, we can figure out that the dwarfing rate was between around 1 kilogram and almost 7 kilograms per generation in terms of weight, and in terms of shoulder height, between 0.17 and 1.26 millimeters per generation. So in millimeters in size, it doesn't look that impressive, but thinking about losing between 1 and 7 kilograms every generation, I find quite remarkable. It's also on the upper end of the estimates for such evolutionary changes in size. So the whole size change seems to have been very fast. However, even so, we couldn't get shotgun sequencing from the Sicilian ones. We actually did get shotgun data, nuclear data from our Neumarkt Nord samples from the straight-tusked elephants from the mainland. And again, we did a nuclear tree like with the other studies, which in this case looked not identical, but comparatively similar to the mitochondrial tree, because in this tree, the straight-tusked elephant is actually the sister group to the African forest elephant, which is still not how it should be according to paleontology, but at least we don't have it interspersed anymore. We have them as sister group. Outgroup to these two guys is the African savanna elephant. Nevertheless, this is a summary tree, and when we looked a bit closer, the picture again became more complicated, because it turned out that part of the straight-tusked elephant genome diverges prior to the divergence of the two African elephant species, followed by several gene flow events, a massive one between Paleoloxodon antiquus and the African forest elephant. And that's why we get the summary tree. And another gene flow event either from the Asian elephant or the mammoth lineage, which might actually explain the morphological similarities. So the straight-tusked elephant was actually a three-way hybrid between an ancient African lineage, the Asian lineage of elephants, and the African forest elephant. Unfortunately, the gene flow seems to have been from the surviving lineages or the mammoths into the Paleoloxodon antiquus. So we don't have any straight-tusked elephant genes surviving in African forest elephants. So whereas we could theoretically breed back a cave bear from our modern brown bears, we can't breed back a straight-tusked elephant from the African forest elephant. So to summarize this part, we can get high coverage genomes if we agree that 15x coverage is a high coverage genome from samples that are more than 100,000 years old, like this straight-tusked elephant. We see that gene flow, we also see that, of course, in modern species, is pervasive in species evolution. It may persist millions of years after the initial divergence, and we also find it in paleogenomes, not only in modern genomes. The straight-tusked elephant was a hybrid species, actually a three-way hybrid species, and I think it was really the first extinct hybrid species we know about. Unfortunately, it looks like its genetic legacy is truly lost. So in that case, we really have a lost lineage rather than a ghost lineage. With that, I would like to thank the people who did the work, my research groups at Leipzig, York, and Potsdam, all funding bodies providing money for the expensive stuff we are doing, and many, many people for providing scientific input and especially for providing the samples without which we couldn't do the work. And thank you everybody for listening. That's the end.
Thank you, Michael, for this really interesting talk. I'm seeing some questions. So the first one, that is also a question that I had, which is the difference between using the nuclear or the mitochondrial DNA?
Well, the mitochondrial DNA is inherited only through the female lineage. So it's a very small separate genome you find in the mitochondria, and the male mitochondria in the sperms are lost during fertilization. So it's strictly inherited through females, and it's a single genetic locus. So it doesn't recombine, which actually means we get only a very small part of the story. The nuclear genome consists of many recombining loci, so it's much more complex. We also get the male side of the history, and overall it's much less influenced by forces like genetic drift, because one part of the nuclear genome might be influenced by genetic drift, but then we have the many other independent loci. So we generally see that mitochondrial DNA as a single locus can give kind of a biased view due to random events.
Okay. We have also a question from Peter Young. What can we do to get better DNA data from non-petrous bone?
Okay, I should say, if it's the Peter Young I'm thinking, I should say hello. Hi, Peter. Long time no see. So what we can do is we can actually do a CT scan, and we found that the hardest parts of bones preserve DNA the best. And funnily enough, for example, for long bones, this is the outermost layer. The part of the bone that people very often remove in ancient DNA research because they fear that it's contaminated. But we found actually that the contamination isn't higher than inside the bone. And due to its density, it has a high endogenous content. It's also known by now that in teeth, one shouldn't take simply the root of the teeth, but one should actually take the cementum, which is again the outer layer of the tooth root close to the tooth crown. So a general outcome seems to be that the denser a part of a bone is, the better it preserves DNA. And the density one can measure with a CT scan without actually damaging the bones. We do that regularly now, that we CT scan our bones in a medical scanner. and determine which is the best region of the bone, because sometimes you also have bones that are modified. So the mineral matrix has been modified in the ground in part of the bone, but it's quite intact in other part of the bone. And that gives us much higher endogenous contents. It's still means that some bones don't work, but at least if we have a collection of bones, it allows us to choose the best bones and the best parts of these bones, and that increases chances substantially.
Okay. We have also a couple of anonymous questions. First of all, could you explain a little bit the progress in sequencing technology regarding paleogenomes, or how many paleogenomes have been sequenced until today?
Oh, well, that depends how you define paleogenomes. If we go to humans, many of the data are actually not full genomes, but they are capture data, which actually means they're SNP data. But if you have 1.5 million SNPs, that gives a fair representation of the genome, especially given how well we understand the nuclear variation in humans. So if we include these data, I think at the moment around 1,500, which is actually quite a lot. If we are talking about true resequencing data, so that we really do shotgun sequencing rather than capture, it's much fewer, and that's also what we use for our mammoths and cave bears, and what most people use who work on animals, for the reason because we know much less about the nuclear variation, so we can't get good capture kits, capture baits for SNPs in these species because we simply don't know so much about the SNP variation. In that case, I don't have really updated numbers, but if we say we take resequenced samples with at least coverage of 1x, then it's anywhere, I think, between 100 and 200 only. So much fewer. Yeah, that's what we have at the moment. But we are catching up with the resequencing data. One, because of the Petros, and B, because sequencing is still getting cheaper, also on a slower rate.
Okay. We have a question from Chris Thomas. Are these non-random examples selected to demonstrate interspecific hybridization, or effectively demonstrate that hybridization is a feature of most species lineage?
Well, I would say I'm not working with enough species to say if it's random or non-random, but I can say that it's a species we have most data in our group, so cave hyenas, cave bears, and elephants. And we started working on them before there was any idea that gene flow would take place, and we detected gene flow in all three of our key species. So in a way, it's non-random because it just turned up in everything. We got enough data to look for it. But I'm pretty sure there are species groups where you don't find evidence of gene flow. But we also know from studies on modern species with modern DNA that gene flow is pretty pervasive, much deeper in evolutionary time than we used to believe. On the other hand, it's also a question, I mean, we define species as something where we couldn't find gene flow with very little data, and now we find gene flow with more data. So it might simply be that gene flow continues for a longer time than we had thought before. At some point, it just ceases. Yeah.
Okay. We have a question from Christian Siadio. I'm not really sure about the first part of the question. I think that he's asking, how can we recognize an ancient DNA, and how do we get ancient samples or bones?
Well, okay, I'll try to interpret the question. How do we recognize if it's ancient DNA? I guess that probably means, how do we know that it's not more than contaminating DNA? And that is actually quite a tricky question. especially for humans, if we work on anatomically modern humans, you can always argue, well, it could be from the excavator, it could be from the person in the lab, because in principle, phylogenetically it looks very similar. Now, there are a couple of ways to distinguish modern contamination from ancient DNA. One is the fragment length, which I mentioned. Modern DNA is on average longer. It's not tremendously long because contaminating DNA comes, for example, from shed skin cells where the DNA is relatively degraded, but nevertheless, it's still longer. What we also see is a pattern of DNA damage that changes the coding, and that is cytosine deamination, because if cytosine gets deaminated, it turns into uracil, and uracil has the same coding properties like thymine. So you get C to T, or on the opposite strand, G to A changes. And we know that these are really pervasive at the ends of the molecules. So we have lots of C to T changes at the ends of the molecules. And we see this pattern in true ancient DNA. Now, if you work with extinct species, there are a couple of other things. Actually, also to some extent with still living species, we can look at the mitochondrial DNA. And we know that mitochondrial DNA in an individual is not variable. So we shouldn't see any variation, any polymorphic positions beyond DNA damage and sequencing errors in mitochondrial DNA. And if we do see quite a bit of that, we know there has been contamination. We can also look at the Y chromosome, provided it's samples from males. And we can look at the nuclear variation. If we see, for example, in humans, you can do that. You can look at triallelic SNPs. There are not very many, but there are some SNPs where you don't only have 2 states, but you have 3 states. And of course, if the DNA is not contaminated, we should always only see 2 states and not 3 different states. There are a couple of things we can actually do. And taking all these together, we can reasonably then estimate how much our data have been affected by contamination. It is definitely the case that we always have some human contamination because all samples have been handled by humans. So the key question is not, are we contamination free? The key question is the contamination low enough that it doesn't affect the biological interpretations? Second part of the question, where do we get samples for ancient DNA from? Well, we work together a lot with paleontologists and archaeologists. We don't dig ourselves. I also have colleagues who occasionally go and dig themselves together with paleontologists. But we really get them through collaborators from university collections, museum collections, and sometimes private collectors.
Okay. And finally, also an anonymous question. What is your next research goal?
Well, usually we're still at a phase where we want to get more data, but specifically because that's still rare for the species we work with. We want to get high coverage genomes for some of the species we work with. So 15x or higher, especially for the cave bears, because there are a couple of analyses like population dynamics through time and heterozygosity estimates that are interesting from an evolutionary perspective that we can only do with high coverage genomes. So one thing is getting high coverage genomes. The other thing is actually looking not only at the population genetics, but also on adaptation. So looking at potentially adaptively relevant differences either over time or between extinct and extant species. And again, for that, we need high coverage genomes.
Thank you. So we don't have any more questions. I just want to thank you, Michael, for your time and effort in answering all the questions. Also, in the name of MDPI and the Genes Journal, thank you really much for joining us today. And I would like to say to everyone again, thank you and stay safe. So if anyone has any other question, we will finish the live session now.
Okay, thanks for handling and for hosting the conference.
Bye-bye.
Bye.